for the invitation. We are going to talk today about gene therapy in Duchenne muscular dystrophy. Maybe I can put my, is it okay that or can you change the slide, please?
На даний час, да. Презентація йде.
Right now, we see your presentation.
So this is my disclosures. I am PI in Pfizer CIFFREO study. We'll talk a little bit about this study. And I was attending Sarepta Ad Hoc Advisory Board, and I'm a PI in exon skipping studies in Israel. Slide, please. Okay, we are talking about gene therapy on actually what is called gene transfer. So what happens is that we take a virus in Duchenne muscular dystrophy. This is AAV virus, adeno-associated virus, and we actually delete the natural viral genes, and inside we put a transcript, which is basically what we want to put is the DNA of dystrophin. Is that okay? The translation is, or should I stop for translation?
Так, шановний професор. Так.
No, we have simultaneous interpretation. Please go ahead. Yes, you can. Yes. Thank you.
Okay, so another slide. So here is the first problem, because the dystrophin gene is very large and it doesn't fit into the small AAV. Therefore, there is production of a mini and actually a microdystrophin. Therefore, the gene therapy in Duchenne does not result in cure, but hopefully will change eventually from severe Duchenne to very mild Becker disease. Slide, please. So when we plan, or these companies plan how to make this injected medicine, it contains three main components. One is the vector, the virus itself. The second one is promoter. Promoter drives expression in what tissue that we want. We want the expression to be in muscle disease. And the third one is the transgene, the DNA itself. And as we'll see, this is a bit different. Each of these components is different in different companies. Slide, please. So what happens is that we inject this treatment IV. It arrives to the muscle cell. It enters into the muscle cell. The viral tissue breaks down, and the DNA enters into the nucleus. In AAV, in Duchenne, the DNA does not incorporate with the human DNA, but creates an episome. So in parallel, there is production of the abnormal dystrophin of the patient and the new dystrophin that we injected. Slide, please. What we'll talk today is about the companies, or what we have so far, preliminary results of human studies in gene therapy. And basically, there are two companies. One is Roche Sarepta, and the second one is Pfizer, who are advanced, and we'll talk about another three other companies that basically that are a little bit behind, but we'll actually go into details in a few minutes. Slide, please. So this is actually how it looks, the construct in these five companies. and you can see that each is different from the other. So the vector is a little bit different. So let's say in Pfizer, the virus is AAV9, while in Sarepta, it's RAVRH74. Also, the promoter is a bit different in different companies, as well as this specific transgene, which means the specific DNA that will get eventually to the muscle. Slide, please. So we'll start and about the results we have so far, actually very preliminary results from all the studies that have been performed. We'll start with Roche Sarepta, and please slide. Another slide, please. Okay, so in Sarepta, there have been so far 4 studies. Next slide, please. Next slide. Okay, so this is phase 1, or the first phase 1 study done by Sarepta. And phase 1 study, actually, the aim is safety, to see how safe is the medicine or what is injected. And actually, this is usually done in a small number of patients. So they did 4 patients. Even though the safety is the aim of the study, we really can see actually some effect. And we can see that actually after 3 years, these 4 children did well. We have to take into account that these are patients, young patients, age 4 to 8. And at this age, anyway, they are doing quite well. And the fact that actually this is a very small number. Next slide, please. Again, next slide, please. Okay, what were the side effects? So the major side effect seen was that half of the patient had significant vomiting early on, and some of them had elevated liver enzyme, which actually all have been resolved. So not major side effect in this trial. Next slide, please. Next slide. Then they went to the phase 2 study. And in this study, they have 41 patients. And what they did is actually what will look in most of the gene therapy study, as gene therapy is just one shot. What they did is half the patient get the actually the medication in the beginning, and half got placebo. And 1 year later, the one who got the medicine will get placebo, and the one who got placebo will get the medicine. So after 2 years, everybody got the medicine, and we get actually data of 1 year of all the patients. And in each year, they have compared to the placebo. Next slide, please. So what did we learn from this preliminary study? First, in terms of safety, actually basically the same. What numbers of transcripts are entering each muscle cell? This is very important. So we can see that after 2 years, in each muscle cell, there was, or in nuclei actually, there was something about 3.4 on average transgene, which is quite nice. And we expect dilution over the time, because there are dilution of the muscle cell over time, replacement. So this is quite nice results. In terms of the protein, we measure in Western blot, we can see that there was something like 40% of normal. Is the protein in the cell membrane? Yes, about almost 80% of the cell is dystrophin stain. And in terms of motor function, what are we doing is the North Star NSAA, which is a motor actually evaluation. And you can see that there was slight improvement. So at least, and we'll show basically in all the studies we have so far, we're talking about preliminary results, there is not a huge change. But if we expect to have even stabilization of the patients, we can see that as a major achievement. Next slide. All right, and then what happened is that Sarepta, after this, moved to another production of the virus. They did it on the lab, then they moved to actually commercial production, and they had to go back to do phase 1. So they did a phase 1 in something like 38 patients, again, looking for safety, different ages. Next slide, please. Again, in terms of safety, basically the same. Vomiting, elevation of transaminases. Next slide, please. What we can see, and this is something that this company presented of the first 11 patients that they had, we can see again that the protein, Western blot, was about 50%. Same in other method, immunofluorescent. Again, almost 4 copies, genome copies in each nuclei. Next slide. And if we look at the clinical endpoint, next slide, please. For this, they did a phase 3 double-blind when they actually recruited 120 patients. Again, half of them getting the injection in the first year, half of them will get it after the second year. This is fully enrolled. We don't have any published or presented data yet from this study, but we know, as I'll inform you, that they did submit the results to the FDA. Next slide, please. We don't have the results of the phase 3 study. What we do have, and this was presented by this company, is integrated results of all the studies before phase 3. And they pulled all the patients together. And what we can see that if we look at the motor outcome, the North Star, we can see that there was, after 1 year, improvement of, on the average, 2.3 points. And what they did, they compared it to external control. What is external control? There are some what we call natural history studies of Duchenne muscular dystrophy. So this is what is expected. Definitely, scientifically, this is not comparable to do a double-blind study, but it gives us a picture what is the course of Duchenne muscular dystrophy, and we can see that we get better outcome. This is also in time to rise from the floor, which was improvement compared to what we expect worsening, and 10-meter walk or run, which there is some improvement with treatment compared to what expected to have actually worsening over time. Next slide, please. So we move from Sarepta to Pfizer, and Pfizer actually had 3 trials. The third one, there's no results yet, it just started. This is actually treating very young people, aged 2 to 4, while other study, the phase 1 was 4 to 12 years old, safety, and actually the phase 3 study is 4 to 7 study. Next slide, please. Okay, this is the phase 1. So in phase 1, again, looking for safety of Pfizer, there was 19 patients. Next slide, please. So what we can see, I'll talk a little bit about efficacy and then side effects, because in Pfizer trial, there were side effects, and we'll talk about it in a minute. So what we can see in the results, if we look at the black, actually in the blue, this is the low dose, and the green, this is a high dose. And this is in the gray what we expect to have, like external control. And we see actually that most patients on gene therapy doing better than what we expect from the natural course of the disease in terms of the motor outcome. Next slide, please. So this is showing the same. We look at a North Star. This is what we expect in 1 year from the control, and this is what we see from the patients in the phase 1 study. Again, few study, early age, but seem to be better results than the general outcome. Next slide, please. We can see that if we go into detail, we can see this in 6-minute walk test. In the elder one, we can see in forced vital capacity, in breathing. In those or a little bit elder age, you can see the pull, the upper extremities strength, and so all the functional assessment 1 year of post-therapy seem, let's say, in very, very preliminary results, seem to be better than in natural history of the disease. Next slide, please. So, therefore, Pfizer went to a phase 3 trial study. Again, the same, 2/3 of the patient in this case will get the treatment in the first year, 1/3 will get it after 1 year. And actually, next slide. So this is a double-blind control study. The primary outcome endpoint will be the North Star, so the clinical outcome. Next slide. However, we have to say that this trial did not yet recruit on patients, so we don't have results. And there were significant adverse or serious adverse events that happened in this trial. First, there was one patient in the non-ambulatory cohort who actually died. And the result of that, the trial was stopped, and then Pfizer stopped dosing this population. In the preliminary, the phase 1 study, there was some acute kidney injury and thrombocytopenia, hemolytic uremic syndrome-like. And after this happened, the steroid dose in the trial was extended, and the dose was actually they given higher dose. And on the, let's say, so far at least in phase 3, we didn't see such actually complication. On the other hand, on phase 3, later on the disease, there were several cases of muscle weakness, myositis, rhabdomyolysis, and 2 cases of myocarditis. So if the first adverse event were related to antibodies to the virus itself, to the AAV, the second in phase 3 is probably related to antibodies to the dystrophy itself. And in most of these cases, they were for specific deletion. And the response to that was, after convening a expert panel, they decided that actually patients with deletions near the N and C terminals will not be treated with gene therapy. Later on, if we'll have some time in the question, we may address this issue. What is the rationale of this decision? So this actually study was stopped. Sarepta restarted dosing in the US, but in Israel, so far, we didn't get the approval to redose the patients. Next slide, please. Next slide, please. So I will talk very briefly now about 3 other companies. One is RegenX Bio, which hopefully will start treatment in the 2023, next year. Next slide, please. And they had unexpected problems in manufacturing, and there was delay in starting the trial. I mean, this is something we see a lot in these trials, that these are very complicated trials, very new one. So delay in beginning the treatment is very common. Next slide, please. Another company is Solid Bioscience. They actually started in 2017. They actually dosed 9 patients, ages 4 to 17. Next slide, please. They also, this is called the Ignite DMD study. They also experienced in 3 of them severe adverse effect. 2 of them were systemic inflammatory response syndrome and 1 thrombocytopenia. This is also the, at a certain point, this was stopped and then restarted. Next slide, please. All the events resolved, but still, Solid Bioscience has decided to stop enrollment in this study. They are now engagement with the FDA how to plan future clinical activities, and there is some rumors the company may be sold to another company and so on. So we don't know at this point what will be the route of this company. Next slide, please. So the last one is Geneton. Geneton is a European, and they started in a different way. They actually, the first part is recruiting 100 patients on a baseline study, just follow-up of the study, and then dosing these specific patients. They had 1 serious adverse event in April 2021. I couldn't find exactly what it is, but it was stopped, and they are planning to start very soon again redosing. Next slide. So summary of all we have. So we have only very preliminary results. All the results we have are provided by this company and presented in scientific meeting, but we don't have manuscripts in journals. So actually, this is not actually had the referees look at them. There is submission of Sarepta to the FDA. Hopefully, if that will pass, we'll have a medication. There are differences between different companies in these 3 components, in the virus, in the promoter, in the transgene. So maybe potentially different in effect and maybe potential difference in safety. So we'll have to look for it. Not all patients may be included in the study, so most study now will not include patients with deletion in the NC terminal. Other patients who have antibodies to AAV, so many patients have natural antibodies to AAV, at this point are excluded from these studies, and the companies are working very hard to try how to clean the antibodies, how to be able really to treat those patients, but it's at this point it's not possible. And treatment actually is associated or may be associated with severe adverse side effects. So this is something that the patients should understand, that there are hope, but still there is some danger in entering at this point of trials of gene therapy. So if we have to summarize this in one patient, we can say that development of AAV gene therapy is still, I think, today the most promising strategy we have for Duchenne muscular dystrophy. But there is some risk, and we are still in the learning curve. So I thank you for your attention. Next slide, please. And I would like to thank all my team who are working with me. As you know, it's not a one-man job, and we have to have a really huge dedicated team to be able to manage and treat a patient with Duchenne muscular dystrophy. And so we have some promising options for the future, and we really hope that these will bring us much better news in future years. Thank you very much.
Дякую, шановний пане професору. І якщо можна, в мене є деякі запитання, чи можна вам задати?
We have a few questions. Can we ask you these questions, dear professor? Dear professor, we have a few questions. Can we ask you?
Шановний пане професору, в нашому
Dear professor, in our Zoom link,
Professor Nevo, you can change translation.
I can change this translation. Let me see.
To English.
Interpretation?
Yes, yes.
English. All right, thank you.
Thank you so much for your speech, dear professor.
Thank you.
Thank you so much for this wonderful presentation of the future, Professor Nevo. And given the fact that most children, most families are waiting to receive the new therapy, modern therapy, and they want to receive more or less specific date or year. In your opinion, how much time will it take for the companies, given that you run these researches and you see how effective these medications are, on what stage they are? So could you tell us when in the world we will have the access to these new medications?
Okay, first, you know, it's very hard always to predict, but the most, or let's say the closest to get actually approval are now Sarepta. So Sarepta and Roche actually submitted to the FDA requests for quick approval of their agent. So if that's the case, we may have approval in something like July or something, or I think something when such request is submitted, there is something like 6 months usually the process of the FDA to give their answer to. I'm not sure what will be the answer of the FDA, if they will approve it or will ask for further information, as we are not aware actually what is the final results of their phase 2/3. I guess they are optimistic because they submitted it. That it is not necessarily the fact that they will actually get the approval. Also, if they get the approval, one of the other obstacles that we are facing is the fact that there will be a huge demand of this medication. And I guess that being an American company, they will start probably in the US. So until we'll get the medication, somewhat later. So maybe a year later, maybe a little bit even more. There's supposed to be production of this AAV and transcripts, etc. And the third one, the third obstacle will be probably the cost of the treatment. We're getting now more and more gene therapies and more very expensive therapies, and in the cost that a regular human being cannot afford that. So this is an issue of government and to get this. In Israel, it's called in the basket, to be included in the basket. But also, let's say if we started with SMA, which was the first disease that actually got a very effective treatment, this was included in the basket. But now, if we'll get more and more of this treatment, it's getting more and more to the countries economic to afford that. But let's hope that I think we'll have to go step by step. I think what we have to hope is that this will be approved and we'll have a medication in let's say, within the next year. And then we'll have to look for all the practical issue, how to get it as fast as we can to our patients.
Дякую за відповідь.
Thank you so much for your answer, dear professor. And there is one more question. Let's talk about the medications that Sarepta company is preparing to produce. What dysfunctions?
Dysfunctions, if you
And
mean
there is no other voice. So I think we lost the connection with the speaker. Sorry.
You lost.
I cannot hear what she's saying. I can hear what you're saying and everybody else, but I cannot hear what the speaker is saying. Something is wrong with her internet connection. So we'll have to wait. Sorry.
Okay.
I'm sorry I had the internet connection, the speaker says. And could you tell us, please, for our audience, because it's not just the doctors who are listening to us, but also patients and families, Sarepta company, what kinds of dysfunctions, what enzyme dysfunctions will it be effective for, so that our listeners would understand who has a chance to receive the fastest treatment?
Dysfunction, I guess the meaning is what we can expect as side effects, right? And the most common side effects we can see, or we saw in Sarepta trial, as I mentioned, were mainly those of vomiting. So many of the patients have vomiting, and some had to be hospitalized and watch for that. And some had actually elevated liver, so some kind of a liver injury, but they were all actually temporary and treated actually very well. The question is that, as we saw with other gene therapies, that what we may expect is we may see immunological side effects that may be related to the virus itself. And we may see actually immunologic effect related to the dystrophy transcript itself. Actually, let's say that these are, even though that we should expect that they may happen, I think still in Sarepta, we've seen actually less than we saw a little bit with Pfizer at this point. And so eventually, even with Pfizer, we think that it's still rare. And the fact that first the steroids now have been increased, and the fact that some of the patients who are more at risk are now excluded from the program, we really hope that we don't have severe side effects. But still, this is something that we'll have to look to see in the future. Hopefully, we'll have, by these management and precautions that are taken at this point, we'll see the effect with, let's say, minimized side effects.
Дякую. І
Thank you.
відповісти на питання.
And could you answer the following question, please. What axon disruptions could be the indication for the use of Sarepta medication?
Great question. I'm not sure I have the list here. Let me see if I can have it. I'm not sure.
Шановні колеги, якщо у вас є запитання, ви
Dear
можете...
colleagues, if you have questions, please write in the chat or raise your hand and we will give you the floor to ask the question.
It's a good question. I don't have it on hand, but we'll actually send you later on the exact.
Да, це питання майбутнього, коли вже буде препарат.
That's the question for the future when this medication is available, of course.
Right. Yeah, but yeah, I mean, there is some, there's some difference between the company which is related to the dystrophy manuscripts. So the thought is, and maybe this is more interesting than the exon itself, even though for the families it's very, very, they want to know actually if their child can be or cannot be included in the study. So we'll try to find out from Sarepta exactly what the exons are. But the idea is that even those who have Duchenne, they don't have zero dystrophy. They have very, very small amount of dystrophy. And if they have this small amount of dystrophy, our body can recognize this dystrophy and does not produce antibodies if we put external dystrophy with exactly the same. But if we have, let's say, Duchenne that misses some of these exons, and these exons are present in the transcript of the gene therapy, there may be immune reaction towards this protein. That's why and because of the transcript has some part of both the beginning and the end, what we call NNC terminal, these are for precaution is actually excluded. This is the idea. I think they have published these exons that are excluded, and we'll try to look for their publication.
Якщо можна, в мене є питання.
I have a question, if I may.
Yes.
Дуже багато питань від батьків стосовно...
There are a lot of questions from parents about vamorolone.
Чи продовжується дослідження по
Is this research on vamorolone continuous about this medication? Because we know that there are fewer side effects in contrast to corticosteroids. And are there patients in Israel who constantly take vamorolone? And what can you say about this medication? And does it help children?
Okay, so we are now moving from gene therapy to vamorolone. So vamorolone is not gene therapy. It's a new type of like steroid, actually. This is actually, I think, also submitted to the FDA. So hopefully this will be a medicine that can be obtained. So far, it is not medication anywhere in the world. We participated in the study of vamorolone. So I think we have 8 patients treated with vamorolone. And in some of them, we could see, I think this has been published. I mean, there are some publication about vamorolone. The advantage of vamorolone is that many of, or some of these patients, first, their height is not limited, so they are not very short, as we can see with other steroids. Some of them do not gain weight. but not all of them. Some of them did get weight. And there are some improvement in bone metabolism in those patients compared to other patients. So yes, we think that vamorolone is a steroid with less side effects that we see with compared to, let's say, prednisone or the Deflazacort if you use the high doses. And we hope that they will be in the market shortly. Yeah, I think hopefully, yes.
Дякую. У мене ще таке
Thank
питання.
you so much. And I have one more question. As a scientist, as a doctor, of course, you prescribe the treatment. And in your opinion, does physical therapy help, and to what extent?
Okay, so let's say what we can expect and what we don't need to expect. Let's say what we don't need to expect from physical therapy. Physical therapy cannot produce strength. So it doesn't counteract the disease itself. But physical therapy in the early stages and hydrotherapy in the later stages, if it's important to counteract or to prevent complication of the disease. Let's say in the beginning, the fact that we can actually prevent contractures, let's say of the Achilles tendon, actually helps the children to walk longer. And this is important. This is very important. As long as the children walk more, they can keep their back straight and actually deterioration of respiratory. So this is important. Also, it is important to have the patient active, not too active, not to do exaggerated work, but being active, walk, being able to do things is important to keep them. And I think this helps the disease. So on the other hand, when, let's say, when patients are non-ambulatory, and then for the lower extremities, you cannot actually counteract the contractions unless they are painful or actually this is debilitating and doesn't help them to sit properly or lie properly. But then the function of upper extremities is important, to work on the upper extremity. In the later stage, definitely physical therapy of the respiratory physical therapy is very, very important. So yes, I think that the treatment or physical therapy is very important. And in addition to that, the physical therapists help us a lot in measuring. So if we need this 6-minute walk test, the North Star, this helps us very much to know the course of the disease and the course of the disease, and help us all the clinical trials to see if our patients are actually improving on this. If you will excuse him, just one last question, please.
Дякую. Дуже дякуємо за вашу участь.
Thank you very much for your participation, for your support, and we hope we'll see you later.
Okay, thank you very much.
Дякую.
Keep you all safe, and let's talk in the meeting next year.